The humoral immune response occurs when a B cell encounters a target antigen. And here the antibodies are specific for that particular antigen are then produced. B cells are activated when the antigens bind to the surface receptors, which cross-links them. And what this does is it's going to trigger a receptor-mediated endocytosis of cross-linked antigen receptor complexes, which would be referred to as the process would be referred to as clonal selection, which is going to lead to the proliferation and differentiation of B cells into effector cells. Most clone cells are going to become plasma cells, which are going to be the ones that are going to produce and secrete the antibody or antibodies. And they can secrete the antibodies at a rate of about 2,000 molecules per second for about 4 to 5 days until they die. And the antibodies will circulate in blood or lymph, and they'll bind to free antigens, marking them for destruction by innate or other adaptive mechanisms. So here we have a diagram showing clonal selection, and in this particular diagram we're looking at a primary response in which the body is exposed for the first time to these antigens. And in this image here we have 3 different B cells, and only one of the B cells has a specific receptor for this specific antigen in the diagram. So the other 2 B cells would not be affected by the presence of these antigens. So those would be in this diagram considered to have non-complementary receptors for those. So what happens here, once they're binded, the antigen is bound to the receptor. That's going to activate the B cells, and that causes proliferation of clones. And some of these clones are going to differentiate into plasma cells, which are a form of the effector B cell, and then they'll secrete these antibodies here. Some of them are going to produce memory cells, which will be ready in case the body's exposed to this particular antigen again later on. So in this case, those would be the memory cells there. They do become, or what they do is they provide immunological memory, and they'll be ready to mount an immediate response to any future exposures. And so here you have a memory B cell in the continuation of this diagram. And in case of a secondary response where you have a subsequent exposure to those antigens, the response is going to be much more rapid and more amplified. So you can see a lot more cells are produced, plasma cells are produced, and producing lots and lots more antibodies. And we're also going to produce, some of these cells are going to produce memory cells as well during the cloning. So in case of the immunological memory, in a primary response, cell proliferation and differentiation upon exposure during the first time, it will take up to 3 to 6 days, which is the lag time there. And then we're going to have peak levels of plasma antibodies reached in 10 days. And then the antibody levels are going to decline. In a secondary immune response here, we're going to have a re-exposure to the same antigen, and that's going to give a faster, more prolonged, and more effective response. And here the sensitized memory cells do provide this immunological memory, and they're able to respond within hours, not days, as the case during the primary response. And here the antibody levels are going to peak in 2 to 3 days instead of 10 days, and at much higher levels. So the antibodies are able to bind with greater affinity, and the antibody level can remain high for weeks, 2 months. And so this graph here shows or illustrates the time and the level of antibodies produced. So here we have some antibody A, and you have a first exposure at time 0. And so you can see the lag time here of 3 to 6 days before you start to see this build up of antibodies. And then the antibodies begin to fall off again. And then during the secondary response to that same antigen later on, in this case on this timeline at day 28, the response is much faster and you get a lot more antibodies produced. And what we're showing here on this other one here is a primary immune response to another different antibody, and you can see that this graph looks pretty much the same as the other one. You're going to have this lag time right here of 3 to 6 days before you start to see a buildup of antibodies in the blood. So active humoral immunity occurs when B cells encounter antigens and produce specific antibodies against them. And so what we're doing here is we're going to be comparing active humoral immunity to a passive. So these are different types of humoral immunity. So there's, looking at active, there's going to be 2 types of active humoral immunity, and this, the first one is naturally acquired. So this is formed in response to an actual bacteria or viral infection. So this would be naturally acquired. And then we have artificially acquired, and this is formed in response to a vaccine. So when you get your vaccines, you're going to be exposing yourself to an attenuated pathogen. And attenuated just means it's been weakened in some way. So the pathogen would not be placed in such a way that it would get you sick. But what it does provide is antigenic determinants when you have these attenuated pathogens, and they are immunogenic and reactive, so they do produce a response there. And what this does, it'll spare the symptoms that occur during the primary response, since you don't have an active form of pathogen being placed in with the vaccine. In passive humoral immunity, your body's not going to work to make any antibodies, so this is passive. And this occurs when ready-made antibodies are introduced into the body. So here your B cells are not challenged by any antigens. And in this case, immunological memory cannot occur. So no occurrence of memory. And protection ends when the antibodies degrade. And there's 2 types of passive humoral immunity. As before, there's a natural and then there's artificial. For naturally acquired, the antibodies are delivered to the fetus via placenta from the mother or to the infant through milk. In artificially acquired, this involves an injection of serum by a doctor or something, and it's going to contain gamma globulins, and this protection is immediate, but it ends when the antibodies naturally degrade in the body. So ends when they do degrade. And this chart here just kind of summarizes your humoral immunity. You have active on the left and passive on the right. For active, you have natural and artificial. And in naturally acquired, this is a case when you actually get an infection by a bacteria or virus. In artificially acquired, this is when you get the vaccine with an attenuated pathogen. And then for passive, again, your B cells are not challenged. And here, in the case of naturally acquired, this will be the case where mom passes antibodies to the fetus or to the infant. And then for artificial acquired, you have an injection of exogenous antibodies like gamma globulin. So looking at antibodies themselves, they're also going to be called immunoglobulins. Let me underline that there. And we're going to abbreviate immunoglobulins, IgS. And these are going to be the proteins that are secreted by the plasma cells. And they're what make up the gamma globulin portion of your blood. So they are capable of binding, these immunoglobulins are capable of binding to specifically with the antigen that was detected by the B cell. Okay, so these immunoglobulins can be grouped into five immunoglobulin classes, and we're going to look at those in a bit. But what are the basic structure of an antibody? The basic structure of an antibody overall is going to be either T or Y-shaped antibody monomer, and there's an image of a monomer to the right over here on our screen, and it's going to consist of 4 looping polypeptide chains linked by disulfide bonds. So those yellow dots that you see right here, that's an example of a disulfide bond, and you can see the loops. And right here you have one light chain and a heavy chain, and on the other side also a light and a heavy chain there. So those are your 4 polypeptides. So the 2 heavy chains are going to be identical, and the 2 light chains are going to be identical. And then you're going to have a variable region, and that variable region is at the end of each arm, and this is the part that's specific for an antigen. So this is where antigens would specifically bind. So here's your antigen binding site, and I'm circling on the light and heavy chain the variable part of the chain, and you see the same thing on the other side as well. Continuing with the structure of antibodies, then the antibody is going to have a stem, which is a constant region, and that stem is right over here. And this area is considered the effector region of the antibody, and this stem is involved in dictating the functions of these antibodies. So there's a common sort of function depending on the type of stem that you have there. Here, this is going to depend on the functioning is related to the type of cell and or chemicals that the antibody can bind to. And it also includes how antibody classes function to eliminate antigens. And here the examples are for some examples. Some antibodies are going to fix complement, others are circulate in blood, and some are going to be found in body secretions. So those things are influenced by the type of stem that we have. This is just a space-filling image of a Y-shaped monomer antibody, and it's color-coded for the different light and heavy chains on there. And we have some classes of these antibodies I mentioned earlier, the 5 major classes. They include the immunoglobulin M, A, D, G, and E. So we're looking at immunoglobulin M first. This immunoglobulin is a pentamer, and it's larger than others, and that means it's made of 5 monomers. This is going to be the first antibody released in response. It is going to be a potent agglutinating agent, which means it can cause clumping with the antigens, and it readily fixes and activates complement. So here we have antibodies that help begin the complement cascade. And then we have immunoglobulin A, and this is referred to as a secretory immunoglobulin. This is a monomer or a dimer, and it's found in mucus and other secretions, which is why it's called a secretory immunoglobulin A. And what this does is it helps prevent the entry of pathogens. There is a table here that helps to illustrate what a pentamer might look like, along with some of the details, and that's true for immunoglobulin M and immunoglobulin A below. So looking at immunoglobulin, a lot of information in the table here is the same that was on the prior slide, but a little bit more detail. Again, it's the first immunoglobulin class secreted by plasma cells during your primary response, and that makes it diagnostically useful. And it readily fixes and activates complement, that was mentioned. And it exists as a monomer and a pentamer. And pentamer means 5, bounded together like you see in the illustration there. The monomer serves as an antigen receptor on the B cell, so the monomer will stay there on the plasma membrane of the B cell and serve as a receptor for that antigen. And the pentamer circulates in the blood, and here we're going to have numerous antigen binding sites, which makes it a potent agglutinating agent. So every one of these areas that are right here are binding sites for a specific antigen. So this one pentamer, large molecule here, can bind to multiple antigens and hang on to them, making sort of a clumping. And then here we have immunoglobulin A, and it is a dimer and referred to as a secretory one, and it's found in body secretions such as saliva, sweat, intestinal juice, and milk. This secretory immunoglobulin is going to help stop pathogens from attaching to epithelial cells surfaces, and the monomer exists in limited amounts in the plasma. And then we have immunoglobulin D, G and E, and immunoglobulin D is a monomer, and it's a monomer, and it attaches to the surface of B cells, and it functions largely as a B cell receptor, so it'll just be present there on the plasma membrane. So we have the plasma membrane here, and there it's going to be a B cell receptor specific for antigens. And then we have immunoglobulin G. This is a monomer, and it's going to make up 75 to 85% of the antibodies in the plasma. So it's the most abundant one. And it's going to be found during the secondary and late primary response to antigens present. And this particular one, the monomers can cross the placental barrier. And then we have immunoglobulin E. This monomer is active in some allergies and parasitic infections, and it causes mast cells and basophils to release histamine, which is involved in inflammation. So this table summarizes, again, some of the attributes of these immunoglobulins. As mentioned earlier, the IgD, the immunoglobulin D, is found on B cell surface and functions as a B cell antigen receptor. Immunoglobulin G, the most abundant antibody in the plasma. It is the main antibody of both secondary and a late primary response, and it readily fixes and activates complement, and it protects against bacteria, viruses, and toxins circulating in the blood and lymph, and it can cross the placenta and confers passive immunity from the mother to the fetus. And then we have immunoglobulin E. Here the stems themselves are going to bend. They're going to bind to mast cells or to basophils, and the antigen binding sites in the receptors and triggers these cells to release histamines and other chemicals that are related to inflammation and allergic reactions. They are secreted by plasma cells in the skin, mucosae, and the gastrointestinal and respiratory tracts, and tonsils. Only traces of immunoglobulin E can be found in the plasma, and levels rise during severe allergic attacks or in chronic parasitic infections of the gastrointestinal tract. We would see a rise in these immunoglobulin E. So these plasma cells actually can switch from making one class of antibodies to another, but that same individual plasma cell is going to retain its specificity for the antigen that it responds to. In this case, in a case of immunoglobulin M, it's released during the primary response, but the plasma cell can switch to immunoglobulin G for the secondary response, and although it's a different class of immunoglobulin, it will still be specific for the specific antigen. So almost all secondary responses are immunoglobulin G, and switching from M to A to E can also occur as well. So antibodies, what they target and how they function, it's important to remember that antibodies here, they do not actually destroy the antigens, but instead they're going to activate and inactivate and tag them. So they can inactivate an antigen and tag them for destruction. So what the antibodies do is they're going to attach to antigens, forming an antigen antibody or immune complex. And there are some mechanisms that the antibodies use during this process. One is neutralization, agglutination, which we've seen before, precipitation, and complement fixation. In neutralization, this is the simplest one here, but it is the most important defensive mechanism. And here what the antibodies do is they neutralize bacteria or virus by blocking very specific sites. So these might be sites on the virus that the virus uses to help enter the host cell. So that blocking prevents binding of those receptors on tissue cells. And the antigen antibody complexes that are produced can undergo phagocytosis and be taken up by phagocytes. And then we have agglutination. And agglutination we've seen before in cases of mismatched blood. Mismatched blood is given during a transfusion. And here the antibodies can bind the same determinant on two different antigens at the same time. And each antibody has two arms, each containing variable region capable of binding to one specific antigen. And so this allows for antigen-antibody complexes to become cross-linked and form a large lattice of clumps. And again, we've seen this when they test for blood types and treat them with specific antibodies. We can get clumping or agglutination. And then there's precipitation. In precipitation here, you're going to have soluble molecules instead of cells. And these soluble molecules are going to bind with the antibodies and cross-link into complexes. These cross-link complexes will precipitate out of the solution. And this precipitated complexes make it easier for phagocytes to engulf them or take them in. And then we have complement fixation and activation here. And this is the main antibody defense against cellular antigens like bacteria. When several antibodies are bound close together on the same antigen, this is going to provide a site for a complement binding site on their stem regions, and because they align in a certain way. This alignment is going to trigger that complement cascade or fixation, which leads to cell lysis and other complement functions, which includes increasing inflammatory response and opsonization for better recognition by phagocytes. And I found an image to kind of show this here. Here you have your plasma membrane, and this is of your antigen, and there's very specific antigens on the plasma membrane right there. And there you can see your antibodies, which are now bounded there. And here you see C1 of the complement system there. And you can see all the series of reactions that are occurring. So just taking you through all the reactions that occur. And ultimately, you get to the C5 and the C6 through 9, which are important in forming the membrane attack complex, or MAC, that is going to ultimately lead to a gap in the plasma membrane that eventually causes the antigen or the cell to lyse like a bacteria cell. And so this diagram here summarizes the mechanisms for antibody action. In the case of neutralization, right here, the antibodies attach to the virus and block it, neutralizing it, preventing it from entering the host cells. And then here we have agglutination, where several cells are bound together. And then we have precipitation, in which insoluble becomes, in which the soluble antigens become soluble, and this allows for better phagocytosis. So all of these three will lead to better phagocytosis. And then we have a complement here where we have several antibodies fixing on the plasma membrane as shown before, and then this allows the complement cascade to occur, producing our membrane attack complex, which leads to lysis of the cells. And this process also helps enhance inflammation and phagocytosis due to the opsonation. And now we end with this homeostatic imbalance for this second one of the chapter here. In the case of parasitic infections like worms, worms are rather large, like Ascaris and another one called schistosoma, and they require a different immune attack strategy. This is because the worms are too big for plan, and plan is the precipitation, lysis, agglutination, or neutralization that we just finished mentioning right now. So what's going to happen here is immunoglobulin E antibodies are going to play a critical role here anyway in the worm's destruction. What they do is they're going to bind to the surface of the worm, making it, marking it for destruction by eosinophils. And so the eosinophils then are going to bind to the exposed stems of the immunoglobulin E, and then this is going to trigger the eosinophils to release their toxic components, which work toward lysing the worms from the outside, so attacking them from the outside. And then here is another interesting technique or technology called monoclonal antibodies. And these monoclonal antibodies are going to be used in clinical in clinical applications and in research. And what these monoclonal antibodies are they're commercially prepared pure antibodies that are specific for a single antigenic determinant. And the way they're produced is by hybridomas, which are cell hybrids that form between the fusion of a tumor cell and a B cell. The tumor cell portion allows the cell to proliferate indefinitely, as tumors can, and B cell portion allows for the production of a single type of antibody. So what you get is a lot of antibody produced here, and these are used again in research, clinical testing, and in cancer treatment as well. So a summary of the antibody actions: the antigen-antibody complexes, they're not going to destroy the antigens, but what they do is they're going to prepare them for destruction by the innate defenses that we started the chapter with. So the antibodies go after extracellular pathogens. They do not invade tissues unless there is a lesion present. A recent exception has been found, and in this case antibodies can actually act intracellularly within the cell if they attach to a virus before it enters, and this appears to activate mechanisms that work towards destroying the virus.